one we definitely need to record. So congratulations on getting part 2 in. How are you guys feeling about them? Not good.
Pretty okay.
Pretty okay. Okay, I'll take that. Great. Anyone else?
Happy that it's done.
Happy that it's done. Fantastic. All right. Well, yeah, I am very much looking forward to grading them. So, as I'm gonna spend the next couple of weeks grading them. It'll take me a little bit to get through them, but in the meantime, as far as class stuff, we are gonna keep moving forward. And so now that we have kind of really tightened up introduction and materials and methods, we can start moving into results and discussion. And so today is an introduction into that. And so on Blackboard this week, as far as your lecture models or lecture modules, we are really diving into experimental design. And so we briefly touched on the principles of experimental design a few weeks ago. And so who can remember the 3 principles of experimental design? What's the first one?
Randomization.
Randomization. Great. Thanks, Steph. What's the second one?
Replication.
Replication. Perfect. Thanks, Tyler. And what's the third one? So we got randomization, we got replication. What's the third one?
Is it local control?
It is local control. Thanks, Tyler. All right. So randomization, replication, local control. So for anytime you see a research scenario, which is how a lot of experimental design is actually taught, it's through the concept of through examples. I want you guys to be thinking of those things. How did the researchers handle replication? How did they handle randomization, and what were their local controls? Because that's going to really inform you as to which experimental designs are even eligible. All right, so what we're going to do first is we're going to take just a really quick tour through Blackboard, make sure everybody's on the same page. Then I'm going to switch gears, and I have kind of an abbreviated lecture for what you guys are going to see this week. And so the reason for that is I wanted to give you some context for where we're headed, but I'm also going to show you how to analyze a data set, because that is a goal that you're going to need to do for this week. And we are going to use those same practices to analyze a portion of the data on the compost study for the individual research projects. And so that is going to help us write the results section for part 3. And so keep in mind that part 3 is due on the 19th, 20th, right? It's the Friday, the 19th, Friday of before Thanksgiving break. All right. And so in part 3, that's where you're going to write up the results section. You're going to generate graphs. You're going to generate tables. So if you fight with Excel like I do, I have to retrain myself every time I use and create tables and stuff. Don't worry, you're not alone. I'm going to help you. And we're also going to do a series of writing workshops to kind of filter that out. And then you're also going to write the discussion piece to your paper as well. So that's where you kind of bring back to the surface all those references that you spent so much time pulling together for part 1 and part 2. That's a really good place to start, is like, okay, these are the results of my study. How do they compare with research that has already been done? And so a good place to start with is with the research that you've already pulled for your introduction section. And then a next step is to go out and actually find other data to help you write your discussion. And so for the next 3 weeks, we are going to spend quite a bit of time in this discussion period, getting ourselves familiar with not only how to analyze data using Excel for the most part, but also how to write it up. All right, so let's go ahead and check in with Blackboard. All right, so we are in week 11, which doesn't seem possible, but it is. And so this week, we are diving off into experimental designs. And experimental designs are really separated out into 2 main categories that we're going to spend a lot of time focusing on. There is more than that. And so the first category that we're going to talk about is single factor designs. And so this week, we are really diving into what a single factor design is. And so you've got a couple of types of note guides available to you. There's also some YouTube videos that are the lecture videos for both a CRD, which is a completely randomized design. There's also an individual video for an ANOVA, which is an abbreviation for analysis of variance. We're going to talk more about CRDs and analysis of variance today. There is an individual video about randomized block designs, which are essentially completely randomized designs, but with one more limit put in place that helps us to account for a different source of variation that could show up in the data. And then the third single factor design that you guys are going to get familiar with this week is what we call a Latin square design. And Latin square designs are really popular in animal science. They're less popular in plant science. And so next week, we're going to talk about a more complex design that is really used heavily in plant science, but it's actually a multi-factor design. It's not a single factor design. All right, and then you guys have 2 things that are due this week. The first is this quiz about single factor designs. Okay, the quiz is going to be a combination of information straight out of the lectures and also some application questions where you are going to have to analyze a data set, where you are going to have to, or sorry, you're probably not analyzing a data set there, but you're going to have to kind of read through a research scenario and identify certain pieces of that research scenario. So that's where things like, what is the independent variable or the treatment group? What is the dependent variable? What is the experimental unit? How did they handle replication, randomization, local control? All of those questions are a really good place to start after you get overwhelmed from reading a research scenario, which will happen, okay? So please just remember that. When you get into that quiz, you're gonna see some problems that are research scenarios, okay? Read through them, take a breath, read through them again, take another breath, and then start asking yourself some questions, okay? So what is the treatment groups? What is the dependent variables or the traits that we're measuring? What are the experimental units? What is how did they handle replication? How did they handle randomization? What are their local controls? All right. The second assignment that is due this week is this analysis of variance quiz. And this is where you guys are going to have the opportunity to actually analyze a data set. And so the data set is actually linked into a Google Sheet. Everybody has the ability to view it, not to edit it. So you should be able to go in and download a copy of it in Excel. Okay, and then today I am going to take you guys through how to run an analysis of variance, both on a PC and on a Mac. Okay, so really quickly, just use your reaction buttons. So raise your hand if you guys are going to be working on a PC. Okay, so we're raising our hands if you guys are working on a PC. Okay. Raise your hand. Go ahead and put your hands down. Raise your hands if you're going to be working on a Mac. Okay. Okay, so as we get going here, I am going to get in on the Zoom room on my Mac and I'm going to share both screens. Okay. So I think we'll run through what it looks like for a PC first, and then we're going to run through what it looks like for a Mac. All righty. Any questions before we get going today? You guys can put your hands down if you want. All right, fantastic. Okay, guys, I am going to share my screen again, and we're going to go through a quick set of slides. And I'm going to try to do Dr. Smith's floating head thing. So we'll see if this works. I finally got it to work last week, so we will see. Of course. Awesome. Yay. Okay. Okay, guys. So we are this week's lecture material is all about experimental designs and specifically single factor experimental designs. All right. So as we start talking about single factor experimental designs, it's going to be really important for us to kind of get an idea of what an experimental design is. And in the most simplest context, an experimental design is where we create a set of procedures to systematically test a hypothesis. So if we think back to basically the end of September, the first part of October in this class, we spent a lot of time developing hypotheses, which were asking a statistical question, right? And the hypothesis is what was going to be tested. The objective statement is how we walked out that testing process, right? And so with the experimental design, these are really important in research because it strategically places a set of procedures or strongholds on an experiment so that we can conduct that experiment with the utmost integrity, accounting for the most variation as possible. Right. So with experimental designs, I don't mean them to sound scary. They aren't. There's a lot of freedom within experimental designs. Our goal as researchers is to keep the experimental design as simple as possible. Okay. So I always employ the KISS method when I talk about experimental designs. So we want to make sure that we keep it simple, right? The simpler our experimental design, the simpler the statistical analysis. So normally, guys, if we were in person or if I was meaner, I would be having you guys hand calculate an analysis of variance. We spend an entire day doing it. And then I have you do it again on your own. And then I have you do it on the final exam because it's really, really, really important that you understand where the numbers are actually coming from in an analysis and how we are generating the probability values or the p-values. But because we're online and I get the feeling that we're pretty stressed, it's in the middle of the semester, I am just going to show you how to do this on the computer, and which is how you're going to do it anyway. Okay, so I am less concerned with you guys knowing where and how we got the numbers, and more concerned with you guys just generating the numbers, and we'll deal with the why on the back end. Also, I tried this last fall. It was a disaster. disaster. So I pulled it out of the curriculum. All right. So when we come up with an experimental design, by keeping our experimental design very simple, that is going to keep the statistical analysis really straightforward, and it's going to help us reduce practical mistakes that can be made when performing the experiment. All right. So if you guys have ever been a part of a research project, or maybe even a part of a group project where you're having to collect data, the less amount of data that you have to collect, the higher the probability is that you are actually going to record the correct numbers. All right. So the more data points that we have to collect, the greater the risk of sacrificing data integrity is. And so that's the same concept with experimental design. All right. So with our experimental design, we are basically breaking them into two major types of designs. There are single factor designs, which we're going to talk about this week. Then there's multiple factor or two factor designs, which we'll jump into next week. Our project, the compost project that we're using for this class, is a single treatment design. All right. So specifically, our project is a single treatment, completely randomized design. Okay, I can't get everything is mirrored and opposite here. That one right there. Okay. And so our basic experimental designs can be completely randomized, a randomized block design, or a Latin square design. And so these three experimental designs are really going to serve as the basis for the majority of the experimental designs that are out there. And more importantly, these designs only have one treatment factor, meaning that we're only testing one hypothesis, okay, or one set of treatments. All right, so this is the part that I want you guys to talk to me about. Relative to the compost study, what are our two treatments? We have just one factor of treatments, but within that level of treatments, we've got two individual treatments. What are they?
Bone in and bone out.
Correct. Bone in is treatment number one. Bone out is treatment number two. All right. So if we had a experimental design that was multi-factor, then we would have at least two levels of treatment factors. And so that would be like if we wanted to expand the project to not only looking at bone in versus bone out, but if we also wanted to look at like stock type, right? So if we were using straw or hay, if we were using bovine carcasses versus pig carcasses or something like that. If we're looking at two major questions, then that means we have more than one treatment factor. All right, so with our three basic experimental designs, CRDs, RBDs, or split plot, the two basic experimental designs, CRD and randomized block design, can also be used for a two treatment factor design. And so these types of designs we're going to get to next week. I just want you to be aware of what they are. All right, so this is where we would look at multiple levels of treatments, okay, where we could test a mineral supplement and a breed effect, or a watering regime and fertilizer levels. All right, so again, we're going to talk about two treatment factors next week. All right, so to go back to our goals for today, it is my goal and it's our objective for you guys to be able to look at a data set and reorganize it and run an analysis of variance on it. And that's our goal today. So if we were going to do this with a single factor, completely randomized design, the research scenario may read something like this. Okay, so researchers are interested in the forage value of individual annual grasses on California rangelands. The objective of the study is to determine forage quality of fillory, vetch, soft chest brome, ripgut brome, and wild oats. Researchers used a plot study where they planted individual seeds in each plot. After the plants reached their vegetative stage and were able to be identified, samples were taken and sent to a lab to determine ADF, NDF, and crude protein. For those of you guys that have been through the nutrition classes, those words are probably ringing some bells. That's how we assess forage value. Okay, so based on this scenario, this is the part where you guys talk to me. What would the independent variables or the treatments be?
The individual forages.
Yes, the species of grasses, right? So filiary, vetch, soft chest, ripgut, and wild oats. Great. So those are our treatments, are the 5 species of grass or 5 species of rangeland plants. What would the dependent variables be?
ADF, NDF, and the crude protein.
Exactly. Thank you, Tim. So ADF, NDF, and crude protein are going to be the traits that we are measuring. All right. And so what is the experimental unit here?
The plots that were individually seeded.
Yep. So individual plot. Great job. All right. So if we were going to set this experiment up out in the field, this is one way that it may look, right? So if this is our entire field here, okay, each color represents a treatment. Okay, so we've got 5 treatments. Okay, and then the way that we're going to handle replication is to make sure that there's at least one plot in each row, one plot of each treatment in each row. Right, so we're going to have 4 replications or 4 plots of each treatment in the entire study. Right, so for example, if filiary is yellow or designated by the yellow boxes here, we've got filiary showing up 1, 2, 3, 4 times. And sorry, I misspoke here. So replication does not mean that we have to have each plot showing up in each row. That was my mistake. That is something for a multi-factor design and a blocked design. All right, if vetch is our purple plots here, you can see we've got vetch in this first row, vetch here in the second, vetch in the third, vetch in the fourth. Okay, so each species type actually shows up in an individual plot. All right, so if we had a small field like out at the university farm, like where Dr. AGRI does his plot trial work, and we went in and we seeded these grasses, and we would do things like mark the plots off, okay, so that they are bordered. We'd make sure that there's room in between the borders. We would make sure that there is room on the outside so we don't get drift and additional seeds popping up. And then one of the local controls that was actually talked about in the scenario back here is this whole concept of after plants reach the vegetative stage, so meaning we're not taking samples until every plot is in the vegetative stage and the plants can be identified. Okay, so those are 2 local controls right there. So plants being identified is really important because we want to make sure that we are collecting a sample of just filiary plants, right? Or just soft chest plants. And so we don't want to get cross-contamination in there because that's going to cause our results to not be true. So this is just one way that we can start to kind of visualize how this experiment is set up. Okay, so this is a completely randomized design where there are 5 treatments and each treatment is replicated 4 times. All right. So the way that we analyze data is or can be done in one of two major ways. Okay, so if we took samples off of that study, so we went out, we did plot cuttings, we looked and made sure that we were sending samples off that was just soft chest and just vetch and just the individual species. Okay, so we did our best there. We sent them to the lab. The lab sent us back results for NDF, ADF, and crude protein. Okay, so that is the data that we're talking about. So we have data for each treatment group. The way that we would analyze that data is one of two major ways if we're using a completely randomized design. We could either do it through a t-test, which you guys may have heard about before in previous classes. They're pretty popular, or a one-way ANOVA, or analysis of variance test. So ANOVA is an acronym for analysis of variance, where we actually assess how much variance is not only within an individual treatment, but between treatments. And the whole idea is so that we can assess how much variation is associated with the data set. So if there is a significant difference between treatment groups, we're able to detect it. And if that's sounding familiar, that goes back to type 1 and type 2 error that we talked about in the statistics section. The cool part about these two analyses, whether we're running a t-test or a one-way ANOVA, is that if we're doing it with a single or completely randomized design, both analyses are going to generate the same results. Okay. As long as it's for a single factor or a single level experiment. All right, so analysis of variance is really, really popular among researchers. It's also broad enough that it can be applied to not only single factor designs, but we can later apply it to multi-factor designs as well. And so as you're reading literature and stuff, you may come across materials and methods sections where they analyze data using analysis of variance. And so that's what I'm going to teach you guys today. All right, so let's look at a different example. Okay, and I'm going to flip back and forth between the slide sets. Actually, I'm going to go through the slide set first, and then I'm going to take you guys through how this actually looks and is executed on Excel, both on a PC and on a Mac. All right, so the overview of this new example is that we know that manganese has an effect on reproductive characteristics and may increase beef cow fertility. Minimal information about manganese blood serum levels exists for cows here in California. So that's kind of our problem. The null hypothesis that we developed was that there will not be a difference in blood serum manganese levels of beef cows grazing at two locations in Northern California. So our independent variable here is location. We have cows that are grazing in Shasta County and cows that are grazing in Butte County. The dependent variable that we're going to look at, we're only going to look at one, is the blood serum manganese levels, and that's going to be expressed in parts per billion. Experimental unit here is individual cow. We handled replication by taking samples from 10 different cows at each location. And we handled randomization because the cows were chosen at random from each location. Some local controls that were put in place for this study that the scenario doesn't tell you, but I'm just telling you because this was one of my projects, was that the cows were grazing similar grasses. They were of similar genetics, and they had this. They had the same mineral supplement available to them. All right, so we analyzed this data using a single factor analysis of variance. Okay, so this is what the data ended up looking like. So we, I don't know where to put my where to put my floating head. Okay. So we went out, collected samples, collected blood samples, did jugular venipuncture. So we were taking the blood from the neck area of these cows. I broke my hand. Lots of good stuff happened. Okay. When we collected this data, we took those blood samples, we sent them off to the lab, and they sent us a file back with the parts per billion of manganese in each sample. So each blood sample was identified by individual cow number and also by the treatment group. And so the treatment group is going to be location in this case. So treatment number 1 is cows in Shasta County. Here's the data. So these are the individual cow IDs, and then this is the manganese level in parts per billion. Treatment number 2 was the North Butte cows, individual cow ID, which really isn't important to us, and the parts per billion of manganese. So that's what our data set looked like. The way that we are going to analyze it is by going through a series of steps. Okay. And so I'm just going to go through these really quickly, and then I'm going to switch over to actually being on the PC, and I'm going to walk you through how to do this step by step. All right, so now that we've got our data set, our whole goal is to generate an ANOVA table. Okay, so we're going to use algorithms that are already in Excel to generate an ANOVA table. And so this ANOVA table is going to have sources of variation over here on the left-hand side. It'll have sums of squares, degrees of freedom, variance, which is also designated by MS. And sorry, I'm using the wrong function of Zoom right now, so everything's in our way. We're also going to generate an F statistic, and then we're going to generate a p-value. Okay, so the p-value is going to be over here to the right-hand side. That's what we're really, really interested in. All right, so for analysis of variance, we are going to first and foremost open an Excel document and to make sure that we have the analysis tool pack. So if you're on a Mac, it's underneath the data menu that's up at the top. It's about part of the way in the center, I think. Go to data and then analysis tools. If it's not there, we can select an add-in, and I'll show you how to do that in just a second. If you're on a PC, you would go to file. If you needed to add it, it would be under the data tab, and it's usually over on the right-hand side and says data analysis. If you don't have it, you can go to file, options, add-ins, analysis tool pack. Once you click that, you can exit out of that window, and you may need to restart Excel for it to show up underneath the data tab. All right, so don't stress over that too much, okay? Stick with me for just a second, and then we'll get into some more specifics about Excel versus Macs. All right, so our first step is to reorganize the data. So the data that we had in this example is going to be reorganized so that the data is in column form. And so all I did was copy and paste them. I created one column that is for the Shasta, the cows that grazed at the Shasta County location, and then a column for cows that were in the Butte location. The next step is to go to our data analysis tab, whether we're on a Mac or a PC, and we're going to select a single factor, sorry, that one right there, single factor ANOVA. After we select the ANOVA single factor, this window is going to pop up. And again, this is on a PC. We're going to look at what it looks like on a Mac here in just a second. All right, so some things that we need to make sure that we are choosing. First of all, our input range is going to be all the data that is contained in those two columns. And I like to include all of it, including the headings. And then I will just check the box labels in the first row. That keeps our results a whole lot cleaner You'll see that here in just a second. I personally like to look at data in columns. That's a personal preference. So that's why I reorganized it into columns. And so if you go with that, you just need to select the columns orientation here. And the alpha value is always going to be at 0.05. So you just need to check and make sure that that's what it is. It is the default in most of these software packages. And then for me personally, I always try to keep the ANOVA analysis with the data set. So I will actually put the ANOVA table into the same sheet that the data is in. And so I'll just go in and select a cell that doesn't have anything in it. All right, so after we select all of that, we're going to hit run, and it's going to generate this ANOVA table for us. Okay, and so we have a set of summary statistics up here at the top, and we've got the ANOVA table down here at the bottom. These summary statistics become really important because it contains the mean, also shown here is the average, that we're going to use when we generate graphs in our study. Okay, it's also going to contain the ANOVA table, and the ANOVA table has some critical information in it. You're going to see this again when you get through the lectures this week. So the ANOVA table is going to use variation between our treatment groups. Okay, so in this case, it would be the variation we see between the cows at Shasta and the cows at Butte. And it's also going to assess the variation that shows up within treatment group. Okay, so within treatment group means that if we look at these cows that were at the North Butte location, they all didn't have the same exact mineral level in their blood. It varied a little bit. And so this within group variation, also known as the error, is going to assess how much variation is within the group. And then I'm not going to go through how we get the sums of squares, degrees of freedom, and variance at this point in time. But what I need you guys to just have some faith in until you can get through the lectures this week is that the p-value is the important piece of information on this ANOVA table. And so this p-value, if you guys have dealt with p-values in previous classes, we have a cutoff level of 0.05. Okay, so our alpha level and our p-value, our alpha level is our cutoff level for our p-value. And so what that means is that if we have a p-value that is less than 0.05, there is a significant difference between the treatments. Therefore, we would reject the null hypothesis. Keep in mind, our null hypothesis that said that there would be no difference between grazing location and cow mineral serum levels. However, if we have a p-value that is greater than 0.05, then no significance was detected between the treatments. Therefore, we fail to reject the null hypothesis. So if you need to write down in your notes, fail to reject is another word used loosely to accept. We accept our null hypothesis. That's fine too. I will crucify you if you put that in a paper though, okay? Because remember, we never accept results of scientific methods, right? They're only supported or not supported based on the constraints of our experiment. Okay, so we never accept anything, nothing is fact, that type of thing. All right, so that is the p-value portion of this. And so you may want to highlight, screenshot this slide because this verbiage becomes really important when we are trying to write up our results section. Okay, so as we move into our last slide here, our again, our p-value is going to tell us if there was a significant difference in mineral concentration or in manganese concentration in the blood between these two treatment group. So our Shasta group is in yellow, our North Butte is in green. And as you can see, one just quick and easy way, it's not foolproof, is that our standard error bars on each of the columns, they actually overlap with one another. So there really isn't any big difference there. And that's what our p-value tells us, because our p-value at 0.058439 is greater than 0.05. Therefore, there's no significant difference between the treatment groups. And so we failed to reject our null hypothesis. Our null hypothesis stated that there would be no difference between treatment groups, and our statistics and our p-value confirm that. Okay, any big questions before I jump over and demonstrate this on Excel? Okay, great. Thanks, Tim. Appreciate that. Okay, guys. So what I'm going to do is show you Excel on a PC, and I'm sorry, we're getting short on time, but I will show you Excel on a Mac as well, if you guys can hang out. Okay. So let's look at our data set. Okay, so this is the data set that we saw from this manganese mineral study. Okay, so again, just to reorient you with this, this is our location. So treatment 1 was Shasta County, treatment 2 was North Butte County. We're going to ignore individual animal ID. And the other piece of information that is really important is the manganese parts per billion. Okay, so what I'm going to do first is I'm going to select this whole entire sheet and I'm going to copy it to a new blank sheet. So step 1 is to reorganize the information, okay, because I like the column form. It's easier for me. So I'm going to make a Shasta column and a North Butte column. And then I'm going to copy. I'm not going to individually transpose numbers or individually move numbers because there's a higher chance that we can transpose something. And we are going to paste the Shasta numbers under the Shasta column, and then the North Butte numbers under the North Butte column. All right, so that takes care of step number 1 there. I think step number 1, I can't remember. The next part is we need to make sure that we have this data analysis tab or data analysis button on our Excel toolbar. Okay, so the way, if it doesn't show up under data, what we can do is go to File, down to Options, and then on the left-hand side of the Excel options window is add-ins, almost to the bottom. We can go to add-ins, and you should get a whole list of add-ins for Excel here. And the one that you want is this analysis tool pack. And once it's highlighted, you can hit OK and get out of the window. And hopefully it shows up in the data tab over here on the right-hand side. If for some reason it doesn't, shut Excel down, save your work, shut Excel down and restart it. And it should show up. All right, so what we're going to do now is, now that we have our data reorganized and we've got our data analysis button here, we're going to go ahead and click on it. And we are going to choose ANOVA single factor. And on our ANOVA single factor, we're first going to choose the input values. And I like to highlight the labels as well as the data here. I'm going to make sure that my data is grouped by columns. There are labels in the first row, my alpha value is always going to be 0.05. And then on the output range, I'm going to choose to just put it a couple columns over where there's nothing on this side of it. All right, so again, we select our input range, make sure the box for columns is checked, check the box labels in first row, and then in output range, you guys can put it on a new sheet if you want. For me, it's easier to stay organized, especially because we're going to eventually look at multiple traits. Just choose a box for output range. Then we hit OK, and we get a summary table that has our means in it, and we get an ANOVA table that tells us our p-value. Okay, so really quickly, guys, our p-value is 0.14. Does that mean we have a significant difference between treatments, or we do not have a significant difference between treatments?
No significant difference.
No significant difference, exactly. Okay, so that is how you get or how you analyze a set of data in Excel. Okay, and we're going to use this for the ANOVA quiz that is due this week. We're also going to use the same procedure for analyzing data from the compost project. Okay, and so I know that we are about out of time. I apologize for that. I'm gonna get into the Zoom room on my Mac, and I'm gonna run through it on the Mac too. Now I just gotta figure out where. Oh, come on, buddy. Sorry, guys, it's gonna take me just a second. Okay. Hey, look, I came into my own meeting. Fantastic. So I need to, on this one, do that. Let me pull up the data. And it's not on there. Got it. Okay. Okay, guys, so now we're looking at a Mac. Things are just a little bit different, but our data is going to be I'm starting with the same file as I did with the PC. So again, step number 1 is I always copy and paste it to a new sheet. And I do that from a data integrity standpoint, as well as just I like clean sheets to look at. Okay. So I always, sheet number 1 for me usually always becomes the master file that doesn't get messed with. If I need to mess with it, I copy and paste it to a new sheet and mess with it on a new sheet. All right, so with the data analysis aspect of this, we're going to go ahead and create our 2 new columns. So Shasta and North Butte. And then I'm going to copy and paste that data into those columns. And then we should be seeing the data analysis button over here under the data tab. If for some reason you don't see that, you can go to analysis tools and just choose the analysis tool pack and hit OK. And the button should pop up. So again, Mac for the win, way less steps, but I'm biased. So, all right. So our next step is then to to analyze the data with an ANOVA. So we'll go ahead and click data analysis, and we're going to go up here to the top, and we're going to choose ANOVA single factor. And very similar to the PC route, we're going to select our input data. Again, the data is organized in columns. That's a preference for me. There are labels in the first row, Shasta and North Butte, and then our alpha level usually always defaults to 0.05, so I'm not going to change that. The output range for me is going to be, I'm just going to put it over off to the side over here. And then we can hit OK, and hopefully this works. There we go. All right, so I love it. Algorithms are different between a PC and a Mac, just a little bit. So our means are the same, but oh no, I think our p-values are the same too. No, they aren't. So the p-value with the, oh my gosh, that's hilarious. The p-value with the Mac is 0.058. It's still high. It's greater than 0.05, so we still do not have any significant difference here. All right, I love algorithms. Everybody does statistics differently. So, okay, so that gives you an idea of how this looks on a Mac. We also did it on a PC. What questions can I answer for you guys? Is anybody completely overwhelmed? Raise your hand. Okay. Thank you for your honesty. This is not the last time we're going to see this. This is recorded. I'm going to get it put up as soon as I get through the next 2 discussion periods. And then if you guys have issues, please grab a bookings appointment with me this week. My week is actually fairly open for a change. So I would love to help you with this one-on-one if needed. And there's lots of tools online as well, if in other YouTube videos that can help you with this as well. But I would rather talk to you. So please don't hesitate to get in touch with me. So, okay. So just as a reminder, we've got the single factor designs quiz due this week, as well as an ANOVA quiz. And to do that ANOVA quiz, you are going to basically do exactly what I just did on either a PC or a Mac, and then you're going to answer some questions about it. So Any questions that I can answer for you about stuff that's due this week, about the part 2, part 3, the project? Anything else? All right, fantastic. Okay, guys, thanks for hanging out. If you want to hang out for the next session, I'm going to do this again. You're more than welcome to. I'm going to start letting the next crew in though, and I'm going to stop recording because I think we are good to go.